Welcome to Virology Live. This is session number 20. Today we're going to talk about antivirals. As we talked about last time, vaccines, of course, can prevent viral disease, but they don't have a lot of effect if you are already infected, with one exception that we've discussed, of course, and that is rabies virus infection. After being bitten by a rabid animal, there's still time to vaccinate you and prevent serious central nervous system effects. But for all other viral infections, the other arm of the defense is antivirals. These can stop an infection once it has started, although the window can be narrow depending on how fast the virus infection is progressing, as we'll see today. We've been working on antiviral drug discovery for over 60 years, but our arsenal is still dangerously small. We only have about 100 or so antiviral drugs on the U.S. market, and most of them, as you can see in this timeline, are against HIV, hepatitis C virus, and herpes viruses. These all, of course, cause persistent infections that go on for a long time, and that should give you a clue about why it's difficult, in part, to make antivirals. That if you have a short, acute infection, you don't have a lot of time to diagnose and then prescribe the drug. Antiviral drug discovery really beginning in the 1960s. And you can see on this timeline, we're going to 2018. And the different curves show you the growth of drugs against different viruses, antiviral drugs. And of course, HIV, we have 41, at least when this slide was made in 2018. There are likely more. We have about 18 for hepatitis C virus. We have some for Hep B, for herpes viruses, and about 9 for influenza virus. Now, obviously, this needs to be updated for SARS-CoV-2. And we'll end up having at least, 3, I suppose, perhaps more. This chart shows you antiviral drugs according to virus and target. So let's start on the left pie chart here. We have 43 antiviral drugs. These are U.S. licensed, FDA licensed, and there are differences in other countries, but keep it simple. 43 against HIV-1, that's the most. 17 against hepatitis C virus. It's a relatively recent development, as we'll see. 8 against hepatitis B. 11 against the herpes viruses. 4 against influenza, and then we have other, not including SARS-CoV-2. This was made in the 5th edition of Principles of Virology. How about targets? These are virus target here, mainly. Polymerase is the biggest one, 27. Protease is 17. Right, so those are the big ones. Then we have some host targets here, 10 host targets. Other viral proteins as targets, NS5A for hepatitis C virus, and the Integrase for HIV-1. And finally, the last pie chart divides it according to virus and host. The vast majority of drugs, and again, these numbers are all numbers of drugs licensed by the FDA in the U.S. These numbers, mostly drugs against the virus and few against the host, although I assume that will continue to grow as we do more work. So why are there so few antiviral drugs? Number of reasons. One of them is that any molecule that interferes with virus growth can also adversely affect the host cell. Why is that? Well, as you know, viruses are parasites, intracellular obligate parasites. They engage many host functions, and so the likelihood that if you inhibit a virus function, it may also inhibit a cell function is high. And such off-target effects, as we call them, if you make a drug against the viral RNA polymerase, it may engage a host polymerase of some kind. We call them off-target effects. They can give you side effects. they can lead to side effects. And that's why we do extensive testing before they're licensed in humans, but sometimes they still remain, and you'll see some examples of that today. Other reasons, well, some medically important viruses, very difficult to propagate, although we've now got around most of that. For many years, it was impossible to grow hepatitis B virus in the laboratory, and the same with human papillomaviruses. And obviously, that doesn't limit you in discovering drugs, because you can always work with the isolated viral proteins, for example. But then at some point you need to measure the inhibition of the drug on virus in cell culture and in animals eventually. And so that can be a problem. Smallpox, very difficult to do as well. Animal models are not really representative of the disease. We have licensed 2 smallpox antivirals in the U.S., and they're stockpiled in the event of a emergence of smallpox. Why would that be? Perhaps bioterrorism, for example. But those 2 were passed, making exceptions to the 2 animal model rule of the FDA needed for licensing drugs. And of course, Ebola virus and Lassa viruses that we need to work on under BSL-4 conditions, makes these kinds of studies challenging because there aren't many BSL-4 laboratories in the world. But a third reason is maybe the most important. Again, a reason why we have so few antiviral drugs, and that is because the compound must block virus replication completely, or as close to completely as possible. In other words, it must be potent. This is not the case for many standard pharmaceuticals. Even if you block partially the activity of some enzyme in you, that's enough to alleviate the signs and symptoms. But partial inhibition is not acceptable for an antiviral. Why is that? Because if you partially inhibit, you will get resistance. It's a good way to get resistant mutants. And this is shown here in this graph where we're looking at viral load on the y-axis and the time after giving the drug. Here, this could be, say, cells in culture. We give the drug, then we infect with these different doses of the same virus. And the optimal dose will completely block virus reproduction, but intermediate and low doses do not. And these allow a certain amount of reproduction, as you can see in the graph, and that will allow for the selection of resistant mutants. This feature makes drug discovery rather expensive. It makes it really expensive because you have to make sure that your drugs are potent. I'm sorry for the pause. I got a text from one of my mods and I thought something might be wrong, so I had to check it. Another serious problem, maybe that interferes with making a lot of antiviral drugs, is that many acute infections are short. As you know from the last time we talked about acute infections, short period of disease, virus onset, and often by the time you feel ill, viral loads are already decreasing. It's too late to impact clinical disease. You have to give, for these short acute infections, the antiviral drugs have to be given early in infection. And this has been a problem for, say, remdesivir for SARS-CoV-2 and COVID. If you give it too late, when viral loads are already declining, then there's little impact on clinical disease outcomes. Now, you could decide to give drugs prophylactically to populations at risk. And this is always a dicey proposition that people don't like to do unless it's necessary. Obviously, there are some exceptions. We do pre-exposure prophylaxis or PrEP for HIV/AIDS. We can take people who have or are at risk for getting infected with HIV-1, and they can do PrEP. We'll talk about that in a bit. But, in general, you don't want to give people anti-influenza virus antivirals all winter in the flu season. Giving drugs to healthy people is, in general, not a good idea. We like to minimize that as much as possible. Now, obviously, for COVID, there are some exceptions. For example, if you had a nursing home with an index case of SARS-CoV-2, you might want to treat the entire population of that nursing home where there are people at high risk for serious disease with an antiviral, if you had such a thing. And now certain monoclonals have been approved for that sort of use. We don't have any broad-spectrum antiviral agents. It's another problem, although I'm going to show you some examples of some candidates which suggest that we could have certain kinds of broad-spectrum antivirals, which then you could use to treat without a diagnosis. But then also, we don't have diagnostic reagents for a lot of viruses. Now, we do have it for influenza. We do have them, obviously, for SARS-CoV-2. But for many acute virus infections, we simply don't test for them. And so you cannot have a virus-specific drug without a virus-specific test. It's kind of like the chicken and egg issue, right? Will the company make a test? No, because there are no drugs. Well, let's make drugs. No, there are no tests. So somebody has to take that first step. Now, obviously, in a pandemic situation, the need drives it. But for many other viruses, it does not. Let's chat a bit about the history of antiviral drug discovery. And as I said before, this search for antivirals started in the 1950s. Of course, by then there had been a lot of success with antibiotics to treat bacterial infections, and so chemists started looking at derivatives of sulfonamide antibiotics to see if they could be used against viruses. Thiosemicarbazones, right here, were found to be active against pox viruses, which of course remained a threat after World War II. And the way this work was done subsequently in the 60s and 70s to expand this effort was by what we call blind screening. You simply look at mixtures of chemicals to see if they have antiviral activity. You simply ask, does this inhibit virus reproduction, say, in cell culture? And if you do find some activity, then you narrow down what is causing that. And again, this is all spurred by our success in treating bacterial infections with antibiotics. So blind screening, a little more on that. There's no attempt to focus on a virus or a specific mechanism, say a target. What you do is you would take random chemicals. So here many companies have huge chemical collections of various chemicals that have been synthesized for different programs over the years. They are sometimes individually stored in vials, or they're stored as mixtures, and they can be screened. You can also, many companies used to go to the soil to take samples of soil and see the microbes that are in the soil if they produce something that is inhibitory to a virus. I used to go, my wife worked at Merck years ago, and when we went on vacation, she would always take soil. This was years ago. And she had a device that would tell her the GPS coordinates. And so if something gave a hit, she could then tell them where exactly it was taken. Of course, you had limitations over what kinds of, you couldn't go on someone's property. Mostly you had to stay on public lands. And so you say, do these inhibit viruses? And then if you get a hit, that is a compound or mixture that blocks viral replication, you then have to purify the active ingredient and see what it is. You have to see if it's, if it has side effects in cells and in animals. And then these molecules can be further modified by the chemists. So they would identify what the molecule is. The chemists would then make changes to it to reduce toxicity, increase solubility, bioavailability, and other pharmacokinetic properties, as we'll see in a moment. And of course, nowadays you can do that kind of screening with mixtures that you buy from companies. I'll get into that in a moment. Now, most of these efforts were rather futile. Thousands and thousands of molecules end up being screened before a specific antiviral even makes it into humans, which should give you some solace if you're worried about safety. Very little success except for one example, Simitrel or amantadine, which was approved in the late 1960s for treating infections with influenza A viruses. That's one of the multiple drugs now available, more than 3, as you'll see in a moment, available for influenza. And we didn't know how it worked for many years. Until the 90s, we didn't know how this worked, and we'll talk about how it works today. We've already encountered the step at which it works. Now, today, antiviral drug discovery is quite different because we have recombinant DNA. We have very sophisticated chemistry, so we can identify a specific target and we can make very specific molecules to inhibit the target based on what the target looks like, the active site of the enzyme, for example. We can clone viral genes and produce the proteins in various organisms, in cells in culture, in yeast or insect cells. We can look at atomic structures and so forth. We know reproduction cycles of most viruses, so we can say what we want to target. And I have to say that the enzymes are a good target because you don't need a lot of inhibitor because the enzyme activities are catalytic, right? And even for viruses that we cannot grow in cells in culture, we can design inhibitors. We could find the polymerase or a protease, make the protein design inhibitors, although at some point you have to be able to test it against virus reproduction. Otherwise, it would be very difficult to end up getting that in people. So we no longer do blind screening procedures. We don't just throw mixtures on cells in culture and infect them with viruses. Well, some people may do that, but that's not the way most drugs end up getting tested. And so, as you know from the first half of this course, we know the reproduction cycles of many viruses. We know there are specific stages to those cycles. We know what goes on. And this slide shows a generic virus reproduction cycle with all the steps that we have, some of which we have touched upon in this course. And we do have drugs that inhibit at each of these stages. We have attachment inhibitors. We have penetration uncoating inhibitors. We have inhibitors of mRNA synthesis, polyprotein processing. Those are inhibitors of proteases. We have many inhibitors of nucleic acid enzymes, both DNA and RNA-based. We have integration and assembly inhibitors, and we have release inhibitors. And so we have covered a good part of the reproduction cycle. And I have to say that our knowledge of this cycle, brought about by research in labs on basic virology, is what informs this work. You need to have the basic science before you can have the clinical advances. The path of drug discovery is long, and this is in part why we don't have antivirals for SARS-CoV-2, or at least many. The three we have were around before the pandemic started, right? We don't have anything yet brand new developed specifically for SARS-CoV-2 that's licensed for use, at least here in the U.S. We will eventually, but the three drugs that we have were discovered before the pandemic. And so, I mean, that's because this is a long pathway, right? You have a medical need identified. You say, okay, we have a virus that causes enough human disease that we need to make an inhibitor. You have to do your research to identify a target. You have to do a proof of principle that if you inhibit that target, it interferes with virus reproduction. So let's say you have a gene in a virus genome and you take it out and it has no effect on reproduction in cells in culture. It doesn't mean that's the end of the line, right? It could have an effect on pathogenesis. So you'd have to do that proof of principle in an animal. If you take the gene out, then you have an altered pathogenesis. But in most cases, deletion of a gene in cells in culture answers the question. And then you have to do some more research, perhaps, to identify the structure of your target, and then start to screen. You develop a screen, and you look at libraries, natural products, compound collections made by combinatorial chemistry, even RNAi, and you do a variety of screens, either cell or mechanism-based. We'll talk about a couple of examples of those in a moment. You can also do in silico screens, where you have the structure, say, of an active site, and you have a computer design potential inhibitors based on the structure of the natural ligand in the site, and that has been done as well. And then you get some hits, which then have to be modified by the chemists to get better compounds, for example. And then at some point, these end up going into animals. You'd make a decision which candidate you're going to pursue, and that's often made on how well it can be synthesized and so forth, as well as its inhibitory and properties and lack of side effects. We did a recent TWIV on the development of Paxlovid, the protease inhibitor for SARS-CoV-2, which is really a nice story about how they went through that. And so you make modifications to the molecules. Will the drug or the compound, it's not a drug yet until it's approved, will the compound get to the right place in the body at the right concentration? Bioavailability. If it's an orally delivered drug, is it going to get where it's needed? Or if not, you may have to deliver it another way, which is not great. Will it persist long enough to be effective? The pharmacokinetic properties, and will it be safe? Toxicity and specificity. And then, armed with all of this, you do tests in animal models to see how well it works against the infection, and then clinical testing in people. A lot of hurdles in the way of finding effective drugs. This is kind of a picture which we made for the principles of virology, just to demonstrate that the numbers are not exactly correct, except the cost, 100, 200 million or more to get a drug to market. 10 to 15 years typically. You make lots of compounds. You reject some of them because there's no antiviral effect. You reject many because they have toxicity. Some don't have viral effect in animals, or they're too toxic in animals. And then you typically pick one to test in humans. There are many shown here being tested in humans, but that may be over the years. If one fails, you would go back and so forth. But you would pick one to test in humans, and by then you have a lot of tox studies in animals. But if it shows in a phase 1 trial toxicity in humans, it would be rejected. And so then you have to show antiviral effect, of course. So that means the agent has to be circulating. And then it gets approved after all of this. So great cost and a lot of time. And here is a summary of drug discovery to the clinic with years here. Typical non-pandemic timeline. Discovery and synthesis of your drug, preclinical work in animals in the laboratory. And then you do a phase 1 trial, typically 20 to 80 healthy subjects. And then those results are typically collected and chewed over for a while. And then you do a phase 2, 100 to 300 efficacy, and then a phase 3, many more subjects. Efficacy, side effects are always monitored, and superiority. If there is already something out there, is this better? Because if there is something and this is not better, it won't be licensed. Then the FDA reviews the data and may approve it for use in human. It's given a license. And then data continues to be collected after the drug is used in humans in order to see, in a larger population, millions of people as opposed to thousands, do we discover anything that we didn't know before. So typically this takes many years to do, and there have been some examples for the COVID pandemic where this has been made much shorter to the detriment of safety. And I'll talk about an example of that in a bit. So let's show you some examples of screens. Here's a mechanism-based screen. So let's say you have a protease and you want to design an inhibitor, a viral protease. And you know the cleavage site for the protease in the protein. So here we made a peptide substrate which incorporates the cleavage site, which is typically between 2 amino acids. And then we make a substrate where we have a bead, a large bead on one end, which simply allows us to remove this from the solution by centrifugation. And then we have a fluorophore, something that makes a signal that we can read on an instrument. And so if you incubate this with the protease, it will cleave this. It will separate the bead conjugated amino acids from the fluorophore in this case. And you can see here's a reaction in time. These are minutes and fluorescence intensity of the soluble peptide. And you remove what's insoluble by centrifuging. That removes whatever's stuck on the bead. And you can see without your inhibitor, there's a lot of activity. And then if you add an inhibitor, it will inhibit this reaction, and you can measure that quite readily. So that's a mechanism-based screen. It is based on looking at, in this case, protease activity, but you could also imagine doing it for polymerase and so forth. And then we have cell-based screens, which utilize cells. In this case, we're utilizing bacteria. And we have a strain of bacteria that are tetracycline resistant. So that's an antibiotic that would normally inhibit the growth of bacteria in culture. And this is a tetracycline resistant multi-transmembrane protein here. It's a pump that will remove tetracycline from the cells so that the cells can grow in the presence of tetracycline. So what you do is you engineer, in this case, the protease site for HIV. And so if you then produce the HIV protease in these bacteria, the protease will cleave this, and that renders the pump inactive. It can no longer remove tetracycline from the bacteria, and so the bacteria will be sensitive to tetracycline. If you plate these bacteria on tetracycline-containing agar, there will be no growth. But let's say if you now add a protease inhibitor to these bacteria, that will, it's shown in red here, will block the activity of the protease. So now these bacteria will be tetracycline resistant because the cleavage site is intact, because the protease is inhibited. So you can do a lot of screening, right? Just screen colonies here, and you can see drugs that give you no colonies versus colonies. And so that is a cell-based screen, as opposed to the previous one, which is done simply in a test tube. So nowadays we do what's called high-throughput screening, kind of like high-throughput sequencing. We can do a lot per day, 10,000 compounds more even. What do we screen? Well, we can screen chemical libraries. You can buy libraries from companies now that are targeted for different sites. For example, you can buy libraries that would inhibit RNA polymerases. You can do natural products as well. You can do combinatorial chemistry. And ways have been devised to take linkers and chemical fragments and mix them in various arrays. You can see here different linkers and different fragments give you many, many different compounds. And the idea being the fragments are binding to your protein, and they're joined together by various chemical linkers. You can do structure-based design if you know the structure of an active site of an enzyme and you know how the substrate fits in. You can design inhibitors based on the substrate. And in silico screening would be tell the computer to tell you what would fit in to that active site and not be and inhibit the enzyme. So high-throughput screening is done largely by robots. It uses microtiter plates like this one with 1,536 wells. And the robots pick up the plates. They add the materials for the reaction, the enzyme, the inhibitor, and so forth. They put them in incubators, and then they put them in readers. And this is all automated, and so you could be sitting at home getting the results of your latest screen while the robots are doing that. So highly efficient, and many, many compounds can be screened. So let's go to our first question. We have many antibiotics, but fewer antivirals. What's the reason for the difference? A, robotic screening is slow. There are few serious viral infections. Resistance is a problem. Antivirals must be potent, or all of the above. And so here's the website you would go. There's a room number you need to get in. It's called Virus. And stay at that web page after you've done the question. Don't close the page. And while you're looking at that, let's have a look at some questions here. Fred asks, can we develop broad spectrum-based diagnostics? Obviously, they tell you which virus is any to solve the chicken and egg. Well, we have a broad spectrum approach, right? You do PCR. You just need to develop the assay. And, I mean, what company will do that if there are no drugs so no doctor will use the assay, right? So I still don't see the solution to that. Yeah, in silico. I like that one. That's a good one. RNA interference. So we talked about RNA interference briefly earlier in the course. It's essentially using short RNAs, 21 nucleotides, that target a gene that you want to inhibit. So you can add those to cells and have your infection, and ask if, say, the target is necessary. You can devise an RNA as an antiviral approach as well. I don't think we will talk about it today, but there is an RNA that can be used for hepatitis C virus. It is directed against the microRNA in the cell that's needed for hep C replication, so this blocks it. It's actually called an antagomer, but they're all based on short RNAs. Explanation of the realities of drug discovery is appreciated. I spent decades in drug discovery, structure-based design, so tired of pharma bashing by those uninformed. Yeah, I mean, Susan, every field gets bashed for some reason by people who don't know about it. But instead of saying you don't know what you're talking about, I would say come and listen to this lecture, right? And see if you can learn something. By the way, these lights behind me, nope, wrong finger. It's a little out of focus, but that is a viral capsid with little lights inside it. Maybe I'll go get it before the end. Could we develop a pan-sarbecovirus? I think so. And I think it could have been done after SARS-1 so that we would have had it ready. But I've said many times I think so. Is the documented antiviral property of ivermectin? Yes, you can ask about it as an antiviral related to cytoplasmic protein tagging and transport. I think it's different for different viruses, and for SARS-CoV-2, which is inhibited in culture by ivermectin, I'm not clear that the mechanism is known. So there are still drugs against chronic HPV in clinical trials. That's correct. Although I think the problem is the drugs don't get rid of the closed circular DNA in cells. That's the problem. You need to get rid of the cell, and the drugs don't do that. What are the three pre-COVID? Remdesivir, molnupiravir, and I actually was including Paxlovid in there. That's actually a drug that was around before, but was extensively modified for COVID. We'll talk about that in a bit. So at least remdesivir and molnupiravir were around before before the pandemic. Would it make sense to screen poop of all fauna for antivirals? That's an interesting approach. So the idea would be that there's some microbe in there that's making an antiviral. That's a good question. It may have been done and it is not useful because soil is a great source. Ivermectin came from a soil streptomyces in Japan originally, right? And many other drugs have come from soils. But that's an interesting one. I don't know the answer to that. It's a good question. Yeah, so they have functional cures for hepatitis B virus. And as I think I mentioned somewhere, when I went to Sweden in 2019, I spoke with an interesting group at the Karolinska where they withdraw antiviral in certain patients. It stimulates virus, it allows virus reproduction, and then you have an immune killing of the infected cells. And so cycles of that, the idea being that you end up killing all the cells with closed circular DNA in them. Yeah. All right. Let's go back to our quiz here. And what do we have? Results. The answer is antivirals must be potent. I mean, many of you answered resistance is a problem. Resistance is a problem, but it's not why we have fewer antivirals. You don't have your resistance already before you make them, right? The Antivirals must be potent is the key one. Screening is not slow. There are a few serious infections. That's not true either. Okay. So only 75%, but I hope some of you got it right. All right, so let's talk about resistance to antiviral drugs. It should be anticipated for any drug that you make. No question about it. Because why? Because viruses reproduce really well, huge levels of virus production, and a lot of mutation at every reproduction. Mutation happens at every reproduction in every cell. It's not something where the newspaper headline says SARS-CoV-2 is mutating. No, it's always mutating. Don't ever make a headline like that, please. And this is of particular concern for long-term infections, persistent or chronic infections like HIV, hepatitis B virus, hepatitis C virus, where you give the drug to patients for a long time and viruses reproducing for long periods, you're going to get resistance. Every HIV drug that we have used, we have resistance to. We will talk about how we got around that. And in fact, we have resistance to every antiviral drug in the arsenal. Now, this slide was made before remdesivir, and so far we don't see remdesivir resistance in people. I would argue that it's not used that much, and it doesn't work all that well in addition. And resistance is a problem because we don't have a lot of drugs, right? If you have resistance to antibiotics or antimicrobials, sometimes you can change and you have a number of alternatives. But what if you don't have another drug for your virus? Then you're in trouble, right? So you can't if you do have resistance, you can't keep treating your patient with the same drug. And if there's no other drug, then the patient's in trouble. You cannot stop the infection. Now, the good part of this, the good news is that you can study resistance. You can say what mutations lead to resistance. That helps you understand the antiviral mechanism and perhaps get around it in some way. Make new strategies to get around the resistance problem. Now, what is the basis for resistance? Error-prone nucleic acid synthesis. For RNA viruses, all polymerases are error-prone, both RNA polymerases and DNA polymerases. And the RNA virus polymerases, like this poliovirus polymerase shown here, for most of them, there's no correction mechanism, right? There's an asterisk here. I would call it the Roger Maris asterisk, and perhaps some of you in the U.S. know what that means. There's no correction mechanism to get rid of the mistakes, so therefore RNA viruses make a lot of mistakes. One misincorporation in 10,000 to 100,000 nucleotides polymerized, which is about 1 million-fold higher than the mutation rate in our DNA genomes. So a mutation rate of, say, 1 in 10 to the 4th misincorporations would lead in a viral genome of 10 kb, one mutation for every genome that's replicated. Or if it's 10 to the 5, it would be in every 10 genomes or so. So the potential is every time an RNA genome is reproduced, you have one mutation introduced. And as you know, there are many, many thousands of reproduction cycles in a single cell. You can make a lot of mutations, right? So here's the asterisk to that statement that, for RNA viruses, there's no error correction. There's one order. So we have an order here. This is a taxonomic name, Nidovirales. An order is just above the family designation. You have all heard of coronavirus families, right, or Coronaviridae. But there are other related families, Arteriviridae, Roniviridae, Mesoniviridae, and these are all put together in an order called Nidovirales. And all these viruses encode a proofreading exonuclease. It's a protein, an enzyme, that sits together with the RNA polymerase. Here's the RNA-dependent RNA polymerase of these Nidovirales in red. NSP14 is the exonuclease. And as the polymerase is polymerizing the genome, making an RNA copy, the complex detects if there are mismatches in the exonuclease, cuts it out so it can be replaced. And so the consequence is that the error frequency of the nidovirales is somewhat lower than that of other RNA viruses, but still higher than for DNA viruses. And these viruses have the largest genomes among RNA viruses, and we think the presence of this exonuclease is a reason for that. In fact, if you can remove the exonuclease gene from the viral genome, and the virus will still reproduce, although it makes more mistakes, and it's not competitive at all with a wild-type virus. And in fact, some have examined that as a potential infectious attenuated vaccine because it doesn't cause disease in animal models. Now, for DNA viruses, DNA polymerases do make mistakes as well, and they have an exonuclease associated with them, so they can excise and replace misincorporated nucleotides. As a consequence, DNA viruses evolve more slowly than RNA viruses, and they also have less diversity, which is not to say that we don't have drug resistance among DNA viruses. We do, but it doesn't emerge as quickly. All right, so now I'd like to go through a set of antivirals and talk about how they work. We're going to go through antivirals that cover pretty much all of the steps of virus reproduction, from attachment and encoding, reproduction and release, and we'll look at some of the licensed antivirals and how they work. Let's start with entry. This is amantadine or Simitrel. This is that one influenza virus antiviral that was discovered in the 60s, and we didn't know how it worked until the 90s. Well, here's the drug. It's an interesting chemistry here. And it interacts with the viral M2 protein. And you may remember, probably not, because it was the first half of this course, we talked about how during entry, the virus particle has an ion channel in it, and that's shown in this diagram right there. We have hemagglutinin, we have neuraminidase. There is the ion channel. And what this does is it allows protons to enter the particle during endocytosis. As the pH of the endosome drops, protons come into the endosomal vesicle. Those protons can then get into the interior of the virus particle, and then the pH drops within the particle. And so when the membrane of the virus and the endosome fuse, which is mediated by conformational changes in the hemagglutinin mediated by the low pH of the endosome, the ribonucleoproteins of the virus can come away from the endosome and get into the nucleus. And so if you inhibit that channel with, say, amantadine, the protons don't get into the virus particle and there's no encoding. So that's where inhibition of influenza virus occurs for amantadine. It's really a beautiful mechanism. Here's a structure of the ion channel in the influenza virus particle. It's a rather short protein. It's a tetramer here to make the channel. Here's the viral membrane. And the protons go right through this channel from the endosome into the virus interior, and that loosens up the interaction of the RNPs, the virus RNA segments, with the M protein that's actually lining the inside of the virus membrane. Now, here's amantadine binding to the channel. It can bind in a number of places. It can bind within the channel to specific amino acids, and it blocks protons from going through. It can also bind on the outside, which apparently causes conformational changes in the channel, and that also blocks the protons from going through. So this we didn't figure out until the 90s, until many years after the drug had been discovered. And unfortunately, most influenza viruses are resistant to amantadine, so we don't use it clinically any longer. That's due to its overuse for many years. All right, so then we have another entry inhibitor, which is Maraviroc, which is this molecule shown here in panel A. And this is a CCR5 inhibitor. So on B is a model of CCR5 in orange, and there's Maraviroc in blue binding to it. Remember, CCR5 is the second receptor needed for HIV entry. Here's the HIV particle with its glycoprotein spikes as trimers. These bind to initially CD4 and then to CCR5, and then the virus will enter cells because the fusion protein gets exposed. In the presence of Maraviroc, the viral glycoprotein cannot bind CCR5, so entry is inhibited. And so this is a drug that targets a host protein, CCR5, and it's really a very nice mechanism. And then we have another drug involved in virus entry, which is may inhibit virus entry, which is hydroxychloroquine. Hydroxychloroquine, of course, licensed as an antimalarial in many countries or derivatives of it. It's known to inhibit infection by multiple viruses by inhibiting endosome acidification. We know, for example, that acidification of the endosome is needed for influenza virus entry, and so hydroxychloroquine prevents acidification of the endosome and it prevents influenza virus reproduction. So early on in the pandemic, it was found that hydroxychloroquine inhibits the reproduction of SARS-CoV-2 in cells in culture. And on the basis of that observation, and the fact that it had been licensed for other uses, it was given an EUA in the U.S., which was not a good decision. This was an example of where the FDA was pushed around and they didn't look at the data. The problem is that this result, inhibition in cells in culture, was done using kidney cells in culture, where the virus does in fact enter through the endosome. The acidification isn't needed for uncoating, but rather the acidification is needed for activation of a endosomal protease called cathepsin L that cleaves the viral glycoprotein and allows it to get into cells by fusion. So you inhibit endosome acidification by hydroxychloroquine. Cathepsin L is not active, and so the virus cannot fuse. The problem is that in the human respiratory epithelium, the virus enters from the plasma membrane because there's a protease called TMPRSS2 that cleaves the spike and then allows fusion at the cell at the plasma membrane. And so hydroxychloroquine has no effect on this entry, and so this drug had no effect in people. And in fact, it caused serious side effects in many COVID patients. There is an inhibitor of TMPRSS2, which, in theory would be tried together with hydroxychloroquine. But that was decided not to be done because HCQ has a lot of side effects. So that's why it failed in people. All right, so let's talk about polymerase inhibitors. There are a lot of drugs that are based on polymerase inhibitors. And the main one is that you take each of the four bases, A, G, C, and T. Right? These are the bases with the sugars attached. They're the ribose ring. And for RNA viruses, of course, it would be, instead of T, it would be a U. And then these are modified chemically. And you can see how a lot of antiviral drugs are modifications of any of the four bases. Here, and the modifications typically inhibit the polymerase, as we'll see. And so let's talk about acyclovir, a very good anti-herpes simplex virus drug. We still use a modified form of it to this day. It's very effective. This is a prodrug, and it's a nucleoside analog. So here's acyclovir. It's made by modifying guanosine. And you can see the ribose is wrecked. So this is still taken, it's still incorporated by the polymerase, but you can't add the next base because there's no hydroxyl here. We'll look at that in a moment. But it's a prodrug. It means it has to be activated in a way, and we'll take a look at that in a moment. But this nucleoside terminology, let's get that straight. A nucleoside has no phosphate. So these are nucleosides in this yellow area. They have no phosphates on them. If you put 1 to 3 phosphates, then it's a nucleotide. And so we have nucleoside analogs of drugs, which would be like these, and then there's some nucleotide analogs, which typically have a single phosphate. Putting phosphates on a drug is tricky because they can't get into cells very well with a phosphate. But there have been some chemical tricks, and we'll have a look at those today. Okay, so acyclovir mechanism of action. The drug gets taken up into cells. Here's acyclovir, no phosphate, so it gets taken up into cells very nicely. And if that cell is infected with herpes simplex virus, the drug is phosphorylated, monophosphorylated by the TK gene product, thymidine kinase gene product of herpes simplex virus type 1. So you put a single phosphate on there. So now you have acyclovir monophosphate, but you need 3. So then cellular kinases add 2 more phosphates. There's 2 of them here, GMP and NDP kinase. So now you have acyclovir triphosphate, which will be utilized by the viral DNA polymerase. It's incorporated. Of course, it's added as a G. So if there's a C in the template, it would be added. But then the next base cannot be added because there's no hydroxyl here on which to add the next base. So it's a chain terminator, and that's how it inhibits the viral DNA polymerase, and it stops virus reproduction. This drug was very good, but it was improved by a chemical modification, which made it higher oral bioavailability. So here is acyclovir, and simply adding valine to it made it so that you could take it orally, it would get into the bloodstream, and then enzymes in the cell cleave off the valine to give you acyclovir. So this is the kind of thing you have to determine empirically, and it works. So it's taken up after oral administration, then acyclovir is released when valine is cleaved off by cellular enzymes. And this is the one that we still use today. Unfortunately, we do get acyclovir-resistant herpes simplex viruses. These arise spontaneously, of course, as the polymerase makes errors. And there are 2 kinds of resistance. There's some where the viruses are resistant because the prodrug can't be phosphorylated. So these changes are in the gene encoding thymidine kinase. So thymidine kinase does not recognize acyclovir. It doesn't put that first phosphate on, and if that's not on, then the second 2 cannot be put on. The other kinds of resistance are in the DNA polymerase. So the DNA polymerase, so TK is fine, we get ACV triphosphate, but then the DNA polymerase does not incorporate it, and so DNA synthesis is not inhibited. And so these are resistant as well. So 2 different categories of resistance because 2 proteins are involved in making this drug work. But the beauty of it is that it is specific for virus-infected cells because only thymidine kinase will phosphorylate acyclovir. So very, very few side effects because it's not active in uninfected cells. Another example of a chain terminator is azidodeoxythymidine, or AZT, which was the first HIV drug. Initially discovered for other purposes, anti-tumor cell compounds. So this was in the freezers of many companies. It was tested early on in the AIDS pandemic and found to inhibit virus reproduction. The way this works, here's AZT here. In fact, we can go back to our original slide, and AZT is right here. So it's a modified form of thymidine, azidotymidine right there. So it is phosphorylated by cellular kinases. There's no viral kinase involved. So here's AZT. It's taken up into the cell, mono, di, and triphosphorylated by different cellular kinases. And then the reverse transcriptase of HIV-1 takes it up and incorporates it. This is a T, right? So it's going to be template to an A. And it acts as a chain terminator. There's no hydroxyl here on which to add the next base. It's a good substrate for HIV reverse transcriptase, less good as a substrate for cell polymerases, so that gives it its specificity, but it still does some inhibition of the cell. And so it has substantial side effects, unlike acyclovir, because it's phosphorylated by cell kinases, and it can get into uninfected cells and inhibit them as well. So it's given orally to absorb rapidly. It's great, but the half-life is just an hour, which is terrible. It's degraded by liver enzymes. Consequently, you have to dose patients 2 to 3 times a day. It has substantial side effects. Many of these patients stop taking their drugs because they feel so poorly. Because the short half-life, multiple dosing regimen, of course, is a formula for getting resistant mutants, and they were selected within a year of the use of this drug. By the way, if you've seen this movie, The Dallas Buyers Club, great discussion of the early days of AZT treatment, the interplay of patients with the FDA, and how patients started sharing their pills, which told the FDA that they could cut the dose in half. It's really quite good. Resistance to AZT arose immediately after the drug was licensed. Single amino acid changes at one of 4 sites in the reverse transcriptase. So here on the right is a model of RT. Polymerase active site, you can see there the yellow active site right there. And the drug, of course, goes in there and is incorporated into the chain and causes chain termination. And there are amino acid changes within the active site lead to resistance. These altered RTs don't bind the phosphorylated AZT, so similar to the DNA polymerase resistance in herpes simplex virus. So rapidly after AZT, other nucleoside analogs were developed, DDI, DDC, D4T, 3TC, of which individually we got resistance, of course. But this led to the idea that we could start doing combination therapy. So people started trying 2 drugs together to treat patients, but still we got drug resistance to 2 drugs in less than a year. And I'm going to give you the numbers for that to show you why in a bit. So the next development for HIV was non-nucleoside reverse transcriptase inhibitors, or NNRTIs. These are drugs that bind away from the active site and induce changes in the protein that cause an inhibition of reverse transcription. They are not chain terminators. So here are 3 of them: nevirapine, delaverdine, and efavirenz. And here, for example, in the red spheres is the binding site for nevirapine. Away from the active site causes an allosteric effect on the protein. So it no longer, allosteric means a change at a distance from the active site. So there's a change, it doesn't work anymore, but it's not a chain terminator. And of course, we got resistance rapidly to these as well. Substitutions in any of the 7 residues that line the binding sites of these drugs. And we don't use them alone any longer. They're used in combination therapy, and that's the format in which they work. Let me tell you about 3 of the SARS-CoV-2 antivirals, and these are nucleoside analogs. Remdesivir, of course, a repurposed drug. It is a prodrug of an adenosine nucleoside analog. And so here's adenosine here. Here's what remdesivir looks like. And it uses a clever chemistry to mask the first phosphate with these chemical groups, which are then removed in cells. That's why it's a prodrug. And then it's already got 1 phosphate on it. So the chemical masking allows this to get into cells effectively rather than if it were an exposed phosphate. And that speeds up the conversion because that first phosphorylation step is often very slow. So it's a chain terminator, and we can't add a base to it. Actually, what happens is it's incorporated, and the polymerase goes a few steps, and then there's a steric clash between the drug, most likely this nitrogen here, and the side chain of the polymerase, and the polymerization stops. So it's a chain terminator, but not because you can't add the next bases, but because there's a clash with the enzyme. This was originally developed for Ebola virus in 2013, so an example of a repurposed antiviral. It does work on multiple RNA viruses. It was found to inhibit SARS-CoV-2 reproduction in cells, and it got a EUA after a couple of trials in the U.S. But it has to be given intravenously, which means that you're often too late. You know, if you wait for people to be so sick that they go in hospital and then they receive this, it's really too late to impact virus reproduction. It doesn't have an effect on hospitalized COVID, although now there have been some trial results suggesting if you give it early enough, it can have an effect, but many people have given up on this. Molnupiravir is the other nucleoside analog. It is a prodrug of cytidine nucleoside analog. So here is cytidine, and here is the actual drug, molnupiravir. You can see there's not many differences here, just this hydroxyl up here. And then this is the prodrug. So there's an extra bit over here that is removed before the drug is activated. Of course, it has to be phosphorylated, and it templates as U. Inhibits multiple RNA viruses. This was known, again, before the pandemic, been developed at Emory University. And then at the pandemic, it was found to inhibit the reproduction of SARS-CoV-2 in cells and in mice. It inhibits reproduction of the virus in ferrets and transmission in ferrets. And so it went through human trials and currently has an EUA. This one is nice because it's orally bioavailable. And we'll see if in fact, we get resistance to this. All right, time for our next question. Number 2. Resistance to which antiviral would involve amino acid changes in a viral enzyme? Acyclovir, amantadine, penicillin, all of the above. A viral enzyme. And while you are looking at that, let's have some questions. And unfortunately, I did forget to mark the last one. Oh, dear. Okay, I picked it up. There aren't so many questions today. Get our ivermectin one. The poop question. Can classification of approaches to antiviral discovery also be described usefully at the level of the Baltimore diagram? I'm not sure it can be, because remember, for the Baltimore is all about genome synthesis and mRNA synthesis. So those are all polymerase issues for the most part. So I think not. Thank you, Susan, for your contribution. Appreciate it. Do you think I would win a Nobel if I came up with a broad spectrum antiviral to kill them all? One drug to kill them all, right? All viruses? Could be. Maybe. You should do it. Yeah, it would be useful, right? If Mother Nature is a mad scientist, then RNA viral reproduction is a drunken orgy in the middle of it. Yeah, it is. They make a lot of changes, and it really drives it. So there was a question here. Can you explain functional treatment? I don't remember where I said that, but essentially treating with a drug that prevents the symptoms, as well as inhibiting replication, because that's how it's working. Although you can imagine you could target some cell protein involved in immune responses. But that's what I mean. Having resistance prevents functional treatment. You can't treat the disease. Why is blind screening the prevalent method? Well, it's not. No, I didn't mean to give that impression. We mostly focus on targets. So you will take a drug like remdesivir and say, does it inhibit? We know already it's a chain terminator for other viruses, so we don't need to do mechanistic-based screens. And then ask, does it inhibit in cell culture? And then we do work to get that licensed. But if you want to discover from start, you're going to pick a target and not just put collections of compounds on cells in culture, infected cells in culture. Oh, this is a good question. In the case of inhibition of CCR5, doesn't that inhibit the function of CCR5 in cells? Yeah, but apparently it doesn't matter. Remember, the 4 to 16% of humans lack the gene for CCR5, and they're fine. So apparently there's some redundancy in whatever it is that CCR5 is doing. So that doesn't make a problem. Good point. Very good point. If we stop using a particular antiviral, will resistance wane as long as there is no fitness advantage? That's right. If you withdraw the drug, well, if the mutation is otherwise neutral, it could be lost spontaneously, right? Of course, if there's a little bit of a fitness cost, it'll be lost quicker. So, yeah, it could go away, but it really depends on the virus and the antiviral. How do these chain terminators not affect the regular cell replication? Well, in the case of acyclovir, right, they can only be phosphorylated in virus-infected cells. An uninfected cell will not phosphorylate it. In the case of the others, they are more efficiently used by the viral reverse transcriptase, which is not to say that there is no incorporation into the cells. So there are side effects, but they're better used by the viral polymerase. Are there examples of heart where all given antivirals are resistant against? Yes. Well, we haven't talked about heart yet, but yes, there are examples. So now when you have a newly diagnosed AIDS patient, you sequence the genome to see what you should give them as an antiviral. And you can find people who have never received triple therapy and they have resistance already because they're out there circulating, yeah. Does acyclovir work similarly in shingles? Yes. Same mechanism, yes. And a number of questions on how they don't affect the cell. And again, they're designed so they're selective for the viral enzyme, but not 100%. It's my favorite virus, my favorite antiviral. So yeah, as Jacob said, my favorite virus is polio. And my favorite antiviral is the one that it's ribavirin that pushes poliovirus over the error threshold. We're going to talk about that next time in, what is next? Evolution. You'll see why. It's very cool. Are antibodies not called antivirals because they're large molecules made by our own bodies? Well, we call antibodies, when you give people antibodies, monoclonal or convalescent serum, right, we call that passive immunization. But they certainly have antiviral effect. So many, yeah, you could say I'm going to give you a monoclonal antiviral. That's fine. But, we try and distinguish between small molecules, which is what we're talking about today pretty much smaller than an antibody molecule as an antiviral. Are those 5% of people without CCR5 immune to HIV? That's correct. How about deleting CCR5? Yes, people are thinking about doing that. We have an HIV lecture where we'll talk about, for sure. Functional cure was used by Tom in the context of chronic HBV where they, yeah. So that's right. Tom said that, not me. So you can treat people with an antiviral. They no longer have hepatitis. That's a functional cure, but the DNA still remains. Thank you, Tom. Very good. All right, let's go back to the quiz now, because today I have a hard out at 1. So I need to be efficient. Show the results. Of course, acyclovir involves amino acid changes in a viral enzyme. Sorry, I need to get rid of this, and I need to go back to the other thing. Here you go. A lot of you picked amantadine, but that's not an enzyme. It's a channel. I tricked you. I know. It's a very nasty question. Penicillin, very good. So it's acyclovir. The viral enzyme is thymidine kinase, right? Amantadine hits a channel. It is not an enzyme. Okay. Integrase inhibitors, we have several of them to block the integration of HIV-1 DNA into the host cell genome. Raltegravir and dolutegravir, RAL and DTG. Remember, integration, retroviral DNA binds the integrase, and then that integrase catalyzes the nicking of host DNA or target DNA, and then ligation of the viral DNA to the host, and that's how the genome integrates. And here we have structures of the active site of the integrase right here with no drug. And you can see here is the viral DNA where it's been cleaved. The 3' hydroxyl is showing, and that's going to be integrated into the host DNA. And the drugs, this is the active site of the integrase. And the drugs, RAL and DTG in magenta and cyan, they fit right in this active site. And they block the integration. In particular, you can see on this diagram there's a drug in there, the magenta, and then there's the target DNA in the host that has to be cleaved, and this is going to just block it from being cleaved. So the integrase inhibitors prevent integration, and they block infection that way because it's an essential part of the reproduction cycle. They're continuing with RNA polymerase inhibitors now. We took a little diversion with the integrase there. Hepatitis C virus RNA polymerase inhibitors have been developed. And here's a diagram of how they work. So normally, as I mentioned briefly before, if you have, say, a nucleoside inhibitor, here's one with a fluorine group here making it an inhibitor. That first step, the addition of the first phosphate, is very slow. And so then the second step, the second and third phosphate additions are faster. But that first one is quite slow. So to improve that, the chemists modified compounds to have already a phosphate in them. And that's shown here in sofosbuvir, which is a prodrug. It's a nucleoside or nucleotide inhibitor now because it has one phosphate onto it. It's got the fluorine there. So it's this one, except it's got chemical modification so that it already has one phosphate, so you don't have to be limited by that first slow phosphorylation step. So now you give the patient sofosbuvir, and then it gets into cells, and this extra stuff is removed by cellular enzyme, and now you have the nucleoside monophosphate in the cell, which can be quickly di- and triphosphorylated, and it will inhibit the polymerase. So the key here was developing this chemistry that would hide the phosphate so the drug gets into cells. Because remember, a naked phosphate will not get into cells because it's negatively charged, and so is the membrane, the plasma membrane of the cell. This is a very good polymerase inhibitor, which has been successfully used. Unfortunately, it's very expensive. It's $1,000 per pill. One pill is $1,000, and a 12-week treatment is $84,000. Now, many countries have negotiated better deals for this, but boy, this is tough. All right, so now also dealing with RNA synthesis, but a different aspect, is baloxavir, which is a recently FDA-licensed influenza virus inhibitor. It's approved for treatment of acute, uncomplicated influenza. People 12 years of age or older who have been symptomatic for no more than 2 days. If you're more than 2 days out, it's not going to work. Not worth taking. So you have a small window. You feel sick, you need to get a test, and then a diagnostic test, and then you have to get your prescription filled. As you can see, it's not easy. Not easy to do. This is an inhibitor of the endonuclease of influenza virus. There it is. That's the drug. Dihydroxy chicken wire, as Dixon would call it, or difluoro chicken wire, I suppose. But remember, the influenza virus enzyme that synthesizes viral mRNAs needs a primer derived from capped cellular mRNAs, and that primer is the target of this drug. So the endonuclease cleaves host cell mRNAs, and that is a viral endonuclease that does that, and this drug inhibits it. So it's a very nice example of how discovery of this endonuclease by basic science led to the identification of a new target for influenza antivirals. So cool, right? All right, moving on to protease inhibitors. The first class, HIV protease, which is absolutely essential for the production of infectious virus. So remember, as the HIV is budding from the plasma membrane, and we talked about this in the assembly session, it's produced in a form that's not infectious because the protease that's inside this particle has to cleave the polyprotein that's in there to give you the final structure of the capsid within the membrane. So that protease does that after the particle has been released from the cell. And if you make changes in the protease, you make non-infectious particles. So it's an essential enzyme, so you can use it as a drug target. And the first really good antivirals, other than the polymerase inhibitors, were designed by looking at the substrate for the protease. So here is the Pol substrate. So remember, one part of the viral genome that's translated into a polyprotein is the Pol substrate, from which the reverse transcriptase and the integrase is cleaved by the protease. And it's cleaved between this phenylalanine and the proline. So chemists looked at it and they said, let's model the transition state just before it's cleaved and then make a drug that's based on the transition state of the substrate. And that was A74702, shown here, and it was moderately active, but not enough. So the chemists modified it and did back and forth with the enzyme inhibition assays, and they finally arrived at the final drug, ritonavir, shown here, which is basically a peptidomimetic. It mimics the protein as it sits in the active site of the protease. Here's the viral protease. Here it's a dimer. There's the active site in yellow, and the drug in red is ritonavir. It sits in the active site. It looks like the peptide, but it sticks in there, and it prevents the bona fide viral protein from getting in, so it inhibits the protease. And this is a peptidomimetic. Many of these inhibitors look like the substrate. It's a small molecule that mimics the peptide substrate. We also have a protease inhibitor for hepatitis C. This viral genome is translated as a polyprotein. It's a long protein from which individual viral proteins are derived by processing. One of the proteases is called NS3, and it's in orange. It cuts all of these bonds here between amino acids to give rise to other viral proteins. And if you inhibit that enzyme, that protease, you would block virus reproduction. And so telaprevir was designed very much like ritonavir for HIV to sit in the active site. Here is the NS3 protease. The active site is in yellow. Telaprevir is in red, sitting in the active site, a peptidomimetic that blocks processing of the viral polyprotein. And that brings us to Paxlovid, which is the SARS-CoV-2 Mpro inhibitor, another protease inhibitor. We talked about this recently on TWIV, the development of this protease inhibitor in terms of what chemistry went on is quite interesting. So here's the coronavirus RNA genome on the left. As you know, it encodes multiple open reading frames. And when this enters the cell, the first open reading frames that are accessed are the 1A and the 1AB protein. They're polyproteins. The rest of the proteins are accessed by subgenomic mRNAs. These polyproteins are processed by the Mpro protease, which is embedded in the protein itself. And these give rise to a variety of proteins needed for RNA synthesis, including the RNA polymerase and the protease itself. So this is a protease essential for virus reproduction. And small molecules were designed to inhibit this, starting with a precursor that had been known for SARS-CoV-1 several years before. That's why I thought earlier all of these SARS-CoV-2 antivirals had been known before. But it was modified extensively to make it more active against SARS-CoV-2. So there's the peptidomimetic. It fits into the active site of the protease. The protease is shown as the gray ribbons here. And here's the protease inhibitor sitting in the active site and blocking proteolysis. And on the right are two graphs which show you the activity of the protease with the inhibitor. So the inhibitor is called PF 073213332. What a great name, right? And this is the rate of the enzyme activity on the substrate. And we're looking at percent velocity versus nanomolar concentration of the inhibitor. And you can see it's a lovely titration. And here we have SARS-CoV-2 in black. And you can look at the 50% inhibitory dose, looks to be just over 10 nanomolar, which is really good. It's a very potent inhibitor. And these are other coronaviruses here, some of which, they have a higher 50% dose, which approaches 100, or even for MERS coronavirus, that's that last one. It's, it's about 1,000, which is not great. You'd probably want to optimize it for MERS. And on the bottom is inhibition of viral reproduction in cell culture by monitoring cytopathic effect, virus-induced CPE here on the y-axis. And here's concentration of the drug. And you can see for SARS-CoV-2, it's the black line. The 50% is, this is for CPE. It's approaching 100 nanomolar, which is really good. And so this, of course, has gone through extensive human testing as well. We also have release inhibitors, and these are the Neuraminidase inhibitors of influenza viruses. So when influenza viruses bud from a cell, of course, the receptor for influenza virus is sialic acid. The virus particle is covered with it, and so is the cell. And so these particles could simply, as soon as they're released, be binding to the same cell and to each other. If it were not for the neuraminidase removes sialic acid from the surface of the cell where the virus is going to bud. Now, many years ago, Peter Palese and colleagues, 1976, this was done just as I had entered Peter's lab, they looked at a mutant of influenza virus where there was a temperature-sensitive change in the neuraminidase. So at high temperatures, 39 degrees Celsius, in infected cells, the neuraminidase didn't work. And you can see the consequences. The virus particles, when released, all stick to each other because they all have sialic acid on them. So this tells us you need neuraminidase to remove sialic acid. So that was the idea for making a neuraminidase inhibitor, which then came to fruition many years later by in silico drug design. They took the structure of the neuraminidase shown here in B and the structure in particular with sialic acid bound, and they made inhibitors that mimic sialic acid. And so here's sialic acid in A, again, the receptor for influenza viruses. And the two initially made were zanamivir and oseltamivir. These were structural mimics of sialic acid. They fit into the active site of the neuraminidase and they inhibit its activity. So they're small molecules that inhibit neuraminidase activity, so the viruses never leave the cell surface, and that blocks virus reproduction. So these inhibitors, designed to mimic the natural ligand of neuraminidase, sialic acid, and the idea was if we can make an inhibitor that looks a lot like sialic acid, then mutations that lead to changes in neuraminidase are not likely to occur because they'll block binding of neuraminidase to sialic acid, right? And then the virus won't be able to work at all. And so that's the idea here. We have sialic acid binding to neuraminidase. Sialic acid is on the host cell. Neuraminidase is the Y, the purple Y. So think of that triangle there. Zanamivir is very close to sialic acid, same shape shown here in this picture. It's very hard to get resistance to zanamivir. Not impossible, but quite hard. Because the idea being if you change the neuraminidase to get resistance to zanamivir, it won't be able to bind sialic acid. On the other hand, oseltamivir is slightly different from sialic acid, as shown by the bell shape here. And so you can get changes in the neuraminidase. These three at 292, 94, and 74 all lead to resistance to Tamiflu because this neuraminidase can still bind sialic acid, maintaining its normal function. So the CDC and many other countries do surveillance for resistance to these anti-influenza virus compounds. And so here, this is for 2019. We're looking at different medications, oseltamivir, peramivir, another neuraminidase inhibitor that's given intravenously, zanamivir. And then our endonuclease inhibitor, baloxavir, they also look for that. These viruses circulating H1N2 and H3N2 are all resistant to amantadine. And so we don't use amantadine anymore. But you can see here they've looked at 2,433 viruses, H1, H3, B viruses. And you can see they're looking at reduced and highly reduced inhibition by oseltamivir. So these are highly resistant here. We got 4. In this case, all H1s. You can see peramivir, we have some resistance. We have fewer for no highly reduced resistance. It's highly reduced activity, mutants that have a highly reduced activity of zanamivir. Again, because this is closer to sialic acid than is oseltamivir. And so far, no resistance to baloxavir, but I'm sure that will change at some point. All right, let's go to our last quiz here. Which of the following targets, which of the following HIV antivirals inhibits the earliest stage of infection? Nucleoside inhibitors, NNRTIs, CCR5 inhibitors, integrase inhibitors, or fusion inhibitors. I didn't actually talk about fusion inhibitors, but I think you understand this mechanistically, so you can get it. And let's go to some questions here. Why not monoclonal T-cell therapy? That's hard because that'd be like a transplant, and then you have to make sure the cells are compatible with the recipient. Not impossible, but more difficult, and that's why. How are most antivirals taken up by cells? So there are for small molecules, there are pumps or channels in the membrane that take up these molecules. They can also be taken up by pinocytosis. Dr. Griffin is constantly stressing the importance of giving antiviral drugs and steroids at the right time. All right, so the steroids are immunosuppressants, right? So you need those if you're in a later phase of COVID where your immune system is causing disease. So you can give steroids then and ameliorate that. However, you don't want to give steroids earlier before these immune symptoms because that would suppress your immune response to the replication of the virus and allow the virus to reproduce uncontrollably. So steroids early are bad. On the other hand, antivirals you need to give early because if, say, you give a patient an antiviral, they come in the hospital because they can't breathe, they're in an immunological phase of the disease. The viral titers are already way down, and an antiviral is not going to help. When antivirals help are shortly at symptom onset or within a day or two, or at the first diagnosis if you are a at-risk patient. Earlier the antivirals, the better. That's the idea. How would HIV antivirals allow COVID to mutate in an HIV-positive person who is not vaccinated? No, so someone with HIV who is untreated, they're immunosuppressed. And so you will have rampant reproduction of SARS-CoV-2. And in fact, vaccines may often not work in an HIV-infected individual or work less well. So that's what's going on there. Jacob, the drug you're thinking about, we're going to talk about this in the last lecture of the course. It is a drug for a spinal disease. It's actually a therapeutic virus. And yeah, it's $1.27 million. It's not a small molecule though, yeah. When these Hep C drugs come off patent, Hep C will become rare. I don't know. I think, a lot of the transmission is now by intravenous drug use, and I'm not sure they're going to take it. I think a vaccine is the way that Hep C is going to be curtailed. How did the chemist discover something that both hides the phosphate and gets removed? So I'm not a chemist, so I'm not going to get all of this right, but there are certain side portions that are known to be removed by cell enzymes. We know the enzymes involved. We can actually make them and test them. So it's a matter of combining that with something that will mask the phosphate. Does taking an antiviral prevent the formation of antibodies? Oh, this is a good question we've talked about a lot on our Q&A with A and V. It depends how early you get it, right? Now, Amy would argue that during that early phase, let's say you get exposed to a virus, it reproduces in you, and then you start to feel like you have a cold and you get a test, and it could be up to a week. The virus is already reproducing in you, and then you get an antiviral. So you should still make a certain number. You should still make antibodies, right? At least some response. So it really depends on how long after exposure you get the antiviral. It's an interesting question, and I'm not sure it's been addressed. It would be really interesting to look at people who have gotten antivirals, and now we can do that with molnupiravir and Paxlovid, and see if they make antibody responses. I think that's a really good question. Thank you, Tom, for your contribution. Glad you like it. So ritonavir turns out to make other drugs more potent. It's not a great protease inhibitor, but it inhibits the cell enzymes that make drugs turn over. So it's often given with other antivirals. Thank you for your contribution. Really appreciate it. I see a great deal of collaboration between biologists and chemists for antiviral production. Absolutely. In companies, the chemists and the biologists have to interact for sure. No doubt about it. What did you say was your most likely candidate for the next pandemic? Well, I didn't. It was Ron Fouché. We had him on a TWIV. I think it was in 2019 when I was in Rotterdam, and he said the H2N2s, which circulated from 57 to 68, population immunity is dropping to them, and he thinks that'll be the next pandemic because it's obviously been hiding out in bird reservoirs, and he thinks that'll be it. Any bad side effects of baloxavir if influenza is not present, i.e., can you take it upon flu-like symptoms? I think so. I think the side effects are minimal. You can do that with the other neuraminidase inhibitors as well, right? If you feel flu-like symptoms in the winter, yes. And as I've mentioned on the Q&A, sometimes that happens. Physicians just prescribe without a test, although you can get it in office, right? So you need a prescription for these drugs, so you're going to have to see a doctor at some point. Do SARS-CoV-2 antivirals work on common cold coronas? Well, there was a slide there for the Paxlovid on some of the common cold. They're less effective. Does the antiviral just slow down the replication? No, remember, it has to be inhibitory. It has to inhibit. Otherwise, talked about this in the beginning, if you have reproduction in, say, suboptimal drug concentrations, that allows resistance to emerge. So you have to be inhibitory, as close to completely inhibitory as possible. Let me see if I can get this. Can we say that the cell will resist the antiviral? That's why the virus will earn this resistance, because it will take whatever the cell can give it. The resistance comes from the virus always, right? Unless it's a cell target. But even then, resistance to CCR5 inhibitors, Maraviroc, comes by changes in the viral envelope protein, not the cell. Thank you, Greg, for your contribution. Appreciate it. Okay, so let us go back to the quiz. Let's see what we got here. So which inhibits the earliest stage of infection would be the CCR5 inhibitors, right? The earliest stage, which is glycoprotein binding to CCR5 is blocked. Everything else is later. Nucleoside, NNRTIs, integrase, it's all later. Fusion is later also, right? Fusion happens after binding. So even though I didn't tell you about fusion inhibitors, you could figure that out, right? Because fusion always happens after binding. All right. How about broad-spectrum antivirals? Are they possible? So a number of years ago, a paper was published showing a compound called LJ001, which had activity by these pluses here against a whole range of RNA and DNA viruses. And the one thing they had in common, the ones that were inhibited, all have envelopes. So the viruses without envelopes, adenovirus, Coxsackie, Rio, they were not inhibited. So this is a drug that trashes membranes. Here it is, LJ001, developed by Ben-Hur Lee. What a great name. He's currently at Mount Sinai, but I did interview him a number of years ago when he was somewhere else. He developed this. And here's what happens when you mix it with VSV, enveloped RNA viruses. Just so you dissolve the drug in DMSO, the virus particles are okay. You dissolve LJ001 in DMSO, it trashes the particle. It breaks the membrane to bits. And another variant of this chemical, which is not active, the viruses are intact. So this drug breaks the membrane of the virus. Why doesn't it affect the cell? Well, because the cell is always able to repair its membrane. Now, this is a broad spectrum, but it's not going any further than this. But it's a proof of concept that we could hit, for example, all enveloped RNA viruses. Now, here's an example of a broad inhibitor of RNA viruses, favipiravir. It's an RNA polymerase inhibitor. Here's favipiravir right here. It's a nucleoside analog. Gets taken up into the cell. It's actually just the base. It gets taken in the cell, and it is given a ribose and a phosphate, and eventually favipiravir triphosphate is made. It's incorporated by the polymerase and acts as a chain terminator. It's active against many viruses, as you can see here. Yes, it was tested against SARS-CoV-2. It is active against SARS-CoV-2. It's also active against minus strand RNA viruses. It's licensed in Japan to treat influenza. So it's an idea that, yeah, you could have a broad spectrum RNA virus inhibitor, but again, you'd need to know if you're infected with an RNA virus. So you still need to have some kind of diagnostics. Here's an example of a broad spectrum inhibitor of DNA viruses. Cidofovir, there it is, nucleoside analog. It's an acyclic cytosine phosphonate. So another trick of hiding the phosphate here by making an acyclic residue. Inhibits all of these DNA viruses. So it's a monophosphate mimic, so it can be rapidly phosphorylated to the di and triphosphate by cell enzymes. And the di form has a higher affinity for DNA polymerases than the host, which is a property in general of these acyclic nucleotide analogs. So in other words, we can do broad-spectrum RNA and DNA. We can do membrane-containing viruses. So far, we haven't done them all, though. Let me end up with two stories of antiviral success. Combination therapy for AIDS and hepatitis C. And remember, these two cause lifelong, persistent infections, giving us plenty of time to develop antivirals. So HAART, of course, highly active antiretroviral therapy, is triple therapy for HIV-infected individuals where we can now treat the infection as a chronic disease. The three drugs target different mechanisms. It's one pill with three inhibitors. Now, it doesn't cure your infection. It's a functional cure. You still have a latent reservoir of proviral infected cells, which you can't get rid of by this, but this suppresses all the symptoms, and you can live your life without dying as long as you take these inhibitors. So here's the mathematics of drug resistance. Let's assume one mutation in the genome is needed for drug resistance. If the mutation rate is 1 in 10,000 bases polymerized, that means every base in the genome is substituted in every 10,000 viruses. If infected people make 10 to the 10th new viruses per day, which is about what has been calculated, that means we will be making 1 million viruses each day with resistance to one drug. So that's why one drug doesn't work. What about two drugs? So the likelihood of developing resistance, if it's 10 to the 4 for one, it's 10 to the 4 times 10 to the 4 for two, or 10 to the 8th. We're still making 100 viruses a day resistant to two drugs. But for three drugs, resistance to three drugs, you need 10 to the 12 viruses per day, and that doesn't happen very often. So it's much, much less likely. And remember, the replication is suppressed to a certain extent by drugs, so this is slowed down a bit. But we do see resistance to all three drugs in some individuals. Viruses are circulating, which just randomly get the three different resistance mutations in them, and they can pop up in individuals. That's why you have to sequence their genomes before you treat them. So here's a list of all the approved anti-HIV-1 drugs. We have the nucleotide inhibitors that we talked about. We have non-nucleoside inhibitors, protease inhibitors, and you also see the year of their development here. We have integrase inhibitors. We have entry inhibitors. We have a fusion inhibitor and the Maraviroc, the CCR5 inhibitor. And then we have combinations. We have 2 and 3 drugs together with all different names made by different companies. So lots of options because this has been infecting people since 1981, at least that we know of. Of course, it's been infecting people probably since the 20s. So we've had a long time to make drugs. Antiretroviral therapy saves lives. These are the number of drugs, the actual and projected numbers of people receiving antiretroviral therapy with years. And the African region is where most HIV/AIDS is, and appropriately they are getting most of the drug, but not quite enough yet. And on the bottom, the number of people dying of AIDS-related causes. We have, in adults, we've averted 10 million deaths so far by treating people with ART. And perhaps importantly, new infections averted due to pregnant mother-to-child transfer or transmission. And so these have been going down as we have prevented them by treating mothers with a dose of a antiretroviral regimen just before birth to reduce the viral load and reduce transmission to the fetus. And now, of course, we also do pre-exposure prophylaxis, daily double therapy, typically tenofovir and emtricitabine. That's Truvada. For those at risk, it reduces the risk of sexual transmission of HIV by over 90%, and intravenous drug use transmission by 70%. And so that's pre-exposure prophylaxis, which was decided to do here, even though it can be an issue. This is a high-risk population, and we do want to cut down the spread. And that's contrasted to post-exposure prophylaxis, of course, which is what HAART is. After someone's positive, then you treat them. Hepatitis C is the other miracle, really. We have shortened the length of treatment and made it possible to eradicate this virus. Now, before 2013, we used to treat patients with pegylated interferon. It's modified interferon to prolong its half-life in the blood, plus ribavirin. And some of the patients had to be treated for up to 80 weeks, anywhere from 25 to 80 weeks. These colors are the length of the shortest and the longest treatment. And then we started to develop direct-acting antivirals, such as boceprevir, telaprevir that we've talked about today. And that went from, but we still had to use ribavirin because we hadn't proven that the others were better. But starting in 2015, we switched to the direct-acting antivirals, as you can see here, double therapy, and that reduced the longest treatment to less than 40 weeks. And as we change the combinations over the years, we can now go down to 12 weeks of treatment for 2 drugs and 3 drugs here. And you can eliminate this infection in a short period of time by double or triple therapy. So really 2 amazing outcomes of antiviral drug therapy. But please remember that we still have 10 to the 16th HIV genomes on Earth today, which I showed you on day 1 of this course. And with this, what does that mean? With this number of genomes, we already have resistance to all the antivirals that we have, that 40-plus list that I showed you, as well as any we will make in our lifetimes or beyond. So we have to be always vigilant about tracking resistance, and making new antivirals, because the HIV/AIDS pandemic is still quite substantial, as we'll see later. So this brings us to our 5 lectures left, or 5 sessions left in this course, which is sad because I'll miss you all, but you know where to find me. Nevertheless, on Wednesday, we'll talk about evolution. Fascinating subject, and start to talk about how viruses change. So let's take some more questions here. Is there a good side to a cytokine storm? Well, you know, the word storm implies that it's not beneficial, right? Making cytokines is necessary for a good innate adaptive response. But if you exceed a certain level, it has to be regulated. So the storm part implies that it's more than you need, yeah. Since many people who have COVID or not vaccinated show up late for antivirals, why isn't there more focus on treatment options for influenza? Well, in certain areas there are. So Daniel Griffin is always talking about later treatment options for inflammation. You know, early in the pandemic, we tried to be laser focused on, say, IL-6, which is a major pro-inflammatory cytokine. And we have inhibitors of IL-6 or its receptor. But that was too narrow. It turns out that Dex is better. Dexamethasone, steroids are better. So we can treat, and many physicians are successful, but we still need a better knife than dexamethasone, I think. Can a person taking heart still transmit? No, the viral loads are very low. No, these individuals typically will not be transmitters. I recall reading about an individual that had full-blown AIDS and recovered without me. Yeah, this is a recent study of someone who recovered spontaneously. In fact, I have to look into that for our last discussion, and I will do that. The last lecture, the last session in this course, second to last, is HIV/AIDS. Thank you, John, for your contribution. Appreciate it. Based on the mathematics of drug resistance, shouldn't the FDA block use of a single antiviral and recommend triple drug? Yeah, but we can't, because to recommend them, they have to be tested together. So we only have testing of monotherapy for SARS-CoV-2 so far, so it can't be done. And the companies have to get together because different companies are making them and they have to test them. And then the FDA could do that. So I agree. I think we're going to see resistance to these monotherapies against SARS-CoV-2. Thank you, Barb, for your contribution. Appreciate it. In theory, wouldn't there be more risks of side effects by using a cocktail of antivirals rather than a single one? Yes, in theory, but they have to be tested as cocktails to make sure that that's not the case. And these combinations, they're not just made up and used. They're given safety testing. Thank you, Kate, for your contribution. Appreciate it. And John, thank you as well. Many of you come back and support all the time. I really appreciate that, because we do have expenses at the incubator, and I want to grow it to involve more people. So it is my dream that we can run a science communication company on donations like yours. It's an experiment. I think it can do it. We'll see. I want it to go beyond me, right? I heard the incubator a lot during TWIVs and some lectures. Yes, so this is our new studio in Manhattan, New York, which Parasites Without Borders, which is Daniel Griffin's nonprofit, and I have leased. It's on 7th Avenue. It has an office area and a recording area, which I'm still building out, so I don't have to do my recordings here in my home basement. I don't mind, but I'd rather be there. Like today, I'm at home for a reason, and my office is full of furniture. So the studio is flexible. I can have guests, and I want to do more there, and I can rearrange it as I wish. It's a large space, and if you're ever in New York, come visit. How do you define drug dose? So you have to define it. I showed you some curves, right? You can define it by the amount of drug you need to inhibit 50% of enzyme activity, or 90% of enzyme activity, which is a higher bar, or 50% of virus reproduction, or 90%. And then you have to test it in animals and get a dose per kilogram, which you then figure out a dose for people, which inhibits virus reproduction. 90% is the best. Thank you, Keith, for your contribution. Really appreciate it. And thank you, Hugo. If you donate $1,000 before the end of the year to Microbe TV or Parasites Without Borders, I'll send you a signed copy of Principles of Virology. Yeah. Thank you, Mr. X. Glad you liked it. And I'll wait here a minute or so to see if some more questions come through. I think it's 10 minutes to the hour. We could go 5 more minutes of questions. If not, we'll say goodbye, but let's see. Do you want to see my my glowing virus? I built this a couple of years ago. It's a very simple thing. It's a terrarium, which you can buy, anywhere. And the door opens here. It's got a little slider. It opens, right? And I just put inside of it, one of those, string lights. It's controlled by this guy here. Isn't that cool? I like it. I think it's great. Turn it on around now. It's better than a Christmas tree, right? Oh, Jacob, no problem. I don't mind answering questions. I like questions. I'm a teacher, right? Why would a teacher not like answering questions? So it's fine. As long as they're relevant. All of yours were relevant. Some people don't ask relevant questions, or they want to be angry at stuff, but I appreciate it. Is a virus living or non-living? Well, go back to lecture 1. Virus particle is not living. The infected cell is living. What is viral entry into the host used for? Well, endocytosis is used to take up nutrients and other materials from the extracellular space. It's an absolute requirement for the cell. In your estimation, is there a realistic hope for an effective antiviral for SARS-CoV-2 before worldwide herd immunity is achieved? Well, we have those 3. I think that's what we're going to have for the foreseeable future, because it's hard to develop more. Some are in development for sure, but I'm not sure they're going to have a major impact. Thank you, Bernard, for your contribution. Oh, thank you so much for your very generous contribution. What's the reason there's no specific antiviral against Zika? Because the outbreak subsided. There are very few cases. And so the motivation went away. Did I mention antiviral drugs in connection with organ transplant? I did not, but that's a big need, because when you get an organ transplant, you get immunosuppressed, right? So you don't reject the organ, and that can bring up herpes viruses in you. So there's a whole field of transplantation infectious disease now that people get ready for those infections. Yeah, for sure. Thank you for these lectures. The more we learn, the better. I know you have a Patreon, but some YouTube channels have something we can join. Yeah, so the problem is YouTube takes a lot, right? Like they take more than Patreon. I mean, I can offer it. I don't mind that. Maybe I will, because when people are here, it's convenient. So thank you for reminding me. I will do that. Oh, yes, the it's an icosahedron, right? So you see the 5-fold axis of symmetry. You can see the 3-fold, the 2-fold. And it's a string light, which is controlled by this thing, yeah. It's no big deal. You just put it inside. But it's a genome. Yes, of course. It is the genome, right? You know of any research dedicated to reactivating latent reservoirs of HIV as part of the strategy? Yeah, definitely. We're going to talk about them in the HIV session, for sure. For sure. I have to go to the dentist Friday, huge campus. Well, you're vaccinated, wear a face mask, you'll be fine. You'll be fine. Why doesn't the RNA DNA replication stop until the host cell dies? I don't know what you mean. It does stop when the host cell dies, for sure, right? Could you define 90% effectiveness? So that's defined in cells in culture or in animals, right? The amount of virus you need, say, to inhibit 90% of cytopathic effect or 90% of enzyme activity. And then you want to pick a drug in mice based on the weight that inhibits virus reproduction and disease specifically. You don't want any disease in the mice, right? And whatever the virus reproduction goes along with that. So inhibition of disease is the key. And it depends on the dose as well and the weight of the mouse. And you extrapolate that to humans. Would you be in love with viruses if you studied in Italy? I don't know. Who knows what would happen if you do different things, right? Can never know. I would hope so. Have you heard of oncogenic viruses associated with parasites? Well, certainly there are viruses of parasites, right? Are you talking about causing cancer in the human host? Don't know. Your message needs to get to more people. I know. So some people, look, this is hard stuff, and I'm never going to get millions of subs, but I do my best, and it's pretty good. All right, here's the last question. Maybe one last one. Is the most single deciding factor if a vaccine against the virus is developed monetary? Well, certainly there have to be enough cases to pay for the vaccine, yeah. So it's not monetary, it's human health. But if there are 10 cases a year, it's not going to be developed because no one can pay for a vaccine. It's going to cost a lot of money to do that. Now, there are some countries that could pay for that, but as you say, there are many countries that do not. And for example, Lassa virus causes many, many deaths a year in Africa. They're not developing a vaccine. Other countries are, which is good. The question is who's going to pay for it. But that's the sort of thing we could do. It's not a huge amount of money. I do think we should be paying for vaccines for many other countries. I happen to join your lectures late. So go to virology.ws/virology-live-2021. And that is the syllabus where you can find the PDFs and the readings. All right, folks, I am going to run because I have a call at 1. Thanks, everybody, for coming. I'll see you Wednesday. We'll be back and talking about evolution. You're great. Thanks, moderators. Thanks, everyone, for coming. Thanks for your questions. Bye-bye.